Hi everyone, thank you for coming. My name is Sean Zhuo. I'm the President and Chief Scientific Officer of Hanker Bio. Today I'm going to spend a little bit of time and try to introduce to you this amazing group and amazing company. Hanker Bio is probably one of the best places I've worked in, and the scientific environment inside this company is just amazing. I find the technology of this Hanker Bio is amazing because we use designer biologics, basically protein engineering, try to optimize and designer biologics to achieve super efficacy as well as to reduce side effect. And the other amazing thing about this company is it's streamlined A to Z operation from discovery to PD-CMC to animal models and to clinical in one shop. This is something very rare to see in the beginning biotech. And the founder, Scott Liu, has been working in this kind of industry for over 30 years. So did I. And the team has over like 10 years of experience. And so from the discovery all the way to clinical. So as such, we have these kind of specialties that we are so efficient that we are able to move a program from 0 to IND clearance in 2.3 years. And today, now I'm going to tell you a little bit about what we do. And we try to sort of engage the immune system synergistically, and try to supplement the shortcomings of PD-1, PD-L1 therapies. We know that since the development of Pembro, Nivo, the immunotherapy has opens up a new chapter for cancer treatment. Okay, and PD-1, PD-L1 therapies has caused a lot of, it's like a revolution for cancer treatment. And a lot of people will benefit from it because this kind of immune checkpoint once worked, it worked in many, many different cancer types. However, the shortcoming is that only about 1/3 of the patient can benefit from these kinds of therapies. And so there are reasons why this kind of PD-1, PD-L1 treatment was not sufficient because people noticed that because of the wrong environment, the lack of memory T cells, and so forth. So what we try to do is we try to incorporate innate immunity into this kind of a therapy scheme. So we know that PD-1, PD-L1 is the target to the adaptive immunity on the T cells. And right now we try to incorporate macrophage into this theme, so we incorporate that like innate immunity. So what we try to do is a macrophage has a receptor called SIRP-alpha. It will interact with the CD47 and send out the do not eat me signal. So what we try to do is we block this signal and let the macrophage to eat tumors. But because macrophage is also antigen presenting cells, so by doing that, it will present the antigen from the tumor into T cells through the T cell receptor. And so that by itself will trigger the activation of T cells. And with the additional blockage of PD-1, PD-L1, we will sort of have a multiple way to trigger the T cells activation. So we will reach sort of a stage that eat me and present me and find me and kill me. So this kind of sequential order, we let the innate and adaptive immunity work in sync. Okay, and this is what we try to do. And because SIRP-alpha CD47 blockage is probably the third one after PD-1, PD-L1, and CTLA-4 immuno checkpoint that shows monotherapy efficacy. So the pathway has been recognized by many big pharmas in recent years. And so I list in these slides a few big deals that has been done in the past 2-3 years. The biggest one is obviously Gilead's acquisition of 47. And what they got is the antibody called magrolimab, which we are going to show the comparison in a moment. And the other one is Pfizer's acquisition of Trillium. And what they try to get is the SIRP-alpha fusion protein. Okay, so these are also the two different approaches to block the signaling pathway. And the problem for these blocking CD47 pathway, although we know that it will trigger phagocytosis against tumor, so that will sort of promote the efficacy. But because CD47 is also everywhere in the body, so that will trigger quite substantial side effect. For example, magrolimab, Gilead's molecule, that will trigger anemia and hemagglutination. And so that's the very obvious side effects. But on the other hand, people try to tone down the efficacy try to save, sort of reduce the side effect. However, but once you reduce side effect, you just cannot see the good efficacy. So it's like one way or the other. So now the question for Hanker Bio is, can we engineer a molecule that is both efficacious and safe? That's our goal. Okay, and today we are going to present to you the data of HCB101. This is our engineered molecule. SERP alpha is a fusion protein fused onto IgG4-Fc. Okay, our molecule looks just like a Trillium's TTI-622. As I mentioned earlier, this molecule was acquired by Pfizer for $2 point something billion. Okay, now we are looking at the data. And so on the top two panels, you're looking at efficacy. The one on the left is the blocking ability between SERP alpha and Rag, which is a cancer cell. And on the right, upper right, is the induced phagocytosis against the cancer cells. So on the top two panels, we're looking at the efficacy. On the bottom two panels, we're looking at side effects, including hemagglutination, as well as on the lower right, phagocytosis against RBC, red blood cells. That's definitely not what we want to see. Okay, our molecule, HCB101, which is an engineered SERP alpha and fused to Fc, is the blue line. And in comparison, magrolimab, anti-CD47 mAb, is shown in green, and the TTI-622, Pfizer's molecule, is shown in red. Without any protein engineering, our molecule should look exactly like the red one. Okay, because of our protein engineering, our efficacy should increase almost 1,000-fold. And if you look at the upper right, in terms of phagocytosis against tumor, it's also amazing. Our efficacy should look just as good as magrolimab. However, if you look down on the lower right, you see magrolimab has caused a lot of issues because of the accidental elimination of red blood cells. So you can see a hike of phagocytosis ability for the green lines. But ours looks just as safe as TTI-622. So from this in vitro data, we have demonstrated that we have a good efficacy and good safety profiles. But how about in real animals? So we move this molecule into exploratory cynotox studies. Okay, in this case, we try to compare our molecule along with the TTI-622. Why? Because of TTI-622, we know it's very, very safe in its safety profiles. We use 50 mg per kg, and our molecule treatment is the blue one. TTI-622 is red. And so if you look at the center panel, the red blood cells as well as platelet, you see our molecule can achieve the same really nice safety profiles in RBC and platelet. So just like TTI-622. So now we know that our molecule is pretty safe. Then the next question is, how about the efficacy? Right? As I just mentioned earlier, the molecules that are safe usually are less efficacious. So now let's look at the 4 different panels. On the left, the 2 panels are the lymphomas, basically they are heme tumors. And the one on the right are the solid tumors. So again, the same colors. Our molecule is blue, and magrolimab is green. TTI-622, Pfizer's molecule, is red. So you can see our molecule on the heme tumor is just efficacious as magrolimab, much better than TTI-622. How about solid tumors? Because we know that anti-CD47 was just not efficacious in solid tumors. So we present two solid tumors. One is small cell lung cancer on the upper right. Okay, in this case, you can see our molecule, the blue one, can shrink cancer pretty well, along with magrolimab. In this tumor model, a TTI-62 just doesn't look efficacious at all. Okay, and on day 25, our molecule is just as good as magrolimab, but however, we notice the two lines start separating. So we decided to give it 2 more doses, and lo and behold, magrolimab cannot hold on to the cancer anymore, so the cancer starts growing up. However, while our molecule HCB101 keeps shrinking the tumor, on day 41, the tumor is gone. So this is just a beautiful, beautiful data. And we thought, hey, maybe we should try different ones. And so we tried WITHER. This is a colorectal cancer. In this case also, TTI-622 and the magrolimab is just not as efficacious. Our molecules just look so good. So up to this point, we have tried 15 different models, 10 in monotherapy and 5 as a combo. And our efficacy just look amazing. So from this data, we need to ask ourselves, what happened to the tumor microenvironment? So we did analysis of what immune cells happen, what happened to the immune cells in the tumor microenvironment. So we analyzed the different kind of cells, and we noticed that in this case, in the WITHER colon cancer, we find that M1 macrophage, which is the tumor-eating macrophage, and M2 macrophage, which is the tumor-protecting macrophage, the ratio between M1 and M2 are higher than other drugs. So this sort of can explain why we can see such a good efficacy with our drug. And so then let's sort of dig even deeper. We try to do the sort of quantitative RNA analysis, try to see exactly what happened to the tumor itself as well as the tumor microenvironment. And in this case, you can see our molecule after treatment, we have triggers a very, very different gene expression than TTI-622 and magrolimab. Okay, so all in all, put together, our molecule is just very unique in the sense that when people say, hi, this is just another CERP alpha fusion, I said no, because we have a quite unique MOAs comparing to the existing therapeutics currently in clinical trials. So we look forward to move this molecule into clinical as quick as possible. And now it's a quick summary of what we have tried. As I mentioned, we tried many different models, including pancreatic, breast cancer, especially this is a sort of a triple negative breast cancer, very hard to treat. Colorectal, small cell lung cancer, lymphoma, AML, NHL. Again and again, our sort of the ORR, or like a tumor suppression index, are much, much higher than TTI-622, which is a Pfizer's molecule, or HU5F9, which is magrolimab, and ALX148. So the final slides that we have achieved our goal, that we want to engineer a molecule that has a very good safety profile, we got it, and also have a very good efficacy, especially in solid tumors, and we also got it. So we have already got IND approved, a clearance from the US FDA, and we are applying for the Taiwan's clinical trials as well. And we look forward to move this molecule into a clinical stage as soon as possible. Thank you for your time and your attention. Thank you.